Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Okay, today we're going to talk about DNA replication. You may also know this as DNA synthesis, especially if you watched a couple videos on the cell cycle. We talk about the S phase, which is for synthesis of DNA, but we're going to call it replication. And synthesis is the same thing as making DNA. So our learning outcome is pretty straightforward. How does DNA replicate it? Now, this is going to be the simplistic version. This would not be for majors, but for a non-major. So I'm going to start out simply, then add a little bit more detail, and then I'm going to, of course, talk about why it's important. You know, I always got to make those connections. So think about this. You know, each of our human cells, there's like 17 trillion of them. Each one of them contains 3.5 billion with a B, as in 3.5 times 10 to the 9 billion base pairs. That's a lot of money, right? So we got 3.5 billion base pairs. Now, you may have heard that term base pair. What does that mean? Okay, think about it this way. We've got these 4 nucleotides: adenine, thymine, guanine, and cytosine. Those are the letters of our genetic code, right? And letters come together to form words. So the sequence of those letters forms words which have meaning. But this base pair, if you recall, let's do a little refresher on some DNA, right? But if you recall, DNA is actually a double helix. Double helix, double. There's 2 strands of DNA held together by weak hydrogen bonds. And those weak hydrogen bonds are easily broken. You experience this every day of your life. Just stick your finger in some water and swirl it around. That's what holds water together. They're not very strong bonds, so they're easily broken. But what I'm trying to say here is the 2 strands of DNA are held together by these weak hydrogen bonds between the nitrogenous bases. So adenine always pairs with thymine, and guanine always pairs with cytosine. So if I have an adenine and thymine paired up together, forming 2 hydrogen bonds, I have a base pair. And we have approximately 3.5 billion of these inside of each one of our cells. That's like 6 feet of DNA inside of each one of our cells, inside the nucleus on top of that. So how much information does 3.5 billion base pairs store? Well, enough to fill about 7,000 of our textbooks. Think about that. I'm showing some cheek cells. Those are actually my cheek cells. I made a swab a few years ago in a laboratory and then took the photo with my iPhone 4. Wow, how dated is that? I should really redo these with my new Samsung. It's got a little bit better camera than the old iPhone 4. But what I'm trying to say is you can see those tiny little nuclei inside those cheek cells. And inside those nuclei is 6 feet of DNA packed up so tightly and perfectly organized, storing the information of about 7,000 of our textbooks. Life is just amazing. You know, cell biology is just amazing. How do our cells replicate our DNA? Well, let's start simply here. It's semi-conservative replication. Wait, semi-conservative? No, they're not making a political statement. They're not part Republican or part Democrat. That doesn't mean anything. What semi-conservative replication means is that each of the 2 strands, we call them the parent strands, forms a template to make daughter strands. So we pull the 2 strands apart and we make the daughter strands from them. Now, of course, what I'm showing you here is incredibly simplified. There are lots of proteins involved with this, something I'll come back to at the end of this. So think about it this way. Here's our 2 nitrogenous bases. We got adenine pairing with thymine, or cytosine pairing with guanine. And remember, 2 hydrogen bonds between adenine and thymine, 3 hydrogen bonds between guanine and cytosine. And those hydrogen bonds are easily broken. That allows you to easily separate the 2 strands. So let me go through this really, really simply. We've got the parent strand of DNA, right? And we pull them apart. So now we actually have 2 parent strands. Then each one of those parent strands is going to serve as a template. And then we're going to make the daughter strands right next to the parent strand. And now the daughter strands are antiparallel to the parent strands. Now, if you remember back with your DNA, DNA has directionality. So now let me get into this directionality a little bit because it does matter. And it also matters a little bit. Well, it matters quite a bit for us, actually. We learned this directionality thing and we quote, oh, they're anti-parallel. One runs 5' to 3'. The other one is running the opposite way. And we don't often know why that's important, but this directionality has a huge implication for eukaryotic cells. Remember, our DNA is linear. It's not connected at the ends. And I'll come back to that. But in this illustration, what you can see is we number the carbons in a DNA molecule. And we number them 1, 2, 3, 4, 5 in the nitrogenous base. And then if you notice the nitrogenous base, which is in purple or blue, there's also the sugars, which are the ribose or the DNA, deoxyribose sugars. They're the beige color. We also number those carbons 1, 2, 3, 4, and 5 prime. And the prime is a designation to separate them from the carbons in the nitrogenous base. So if you look at these strands carefully, you'll notice like the phosphate, that little P with a yellow circle around it, on one side they're pointed down and on the other side they're pointed up. If they're pointed up, that's a 5' direction. And then that little hydroxyl group, that OH is at the bottom, that's a 3'. And what that's showing is a 3' carbon. In this, you can also see the nitrogenous bases pairing with each other, forming the hydrogen bonds. Going back to a more simplistic view, you can see the two parent strands in light blue, and you can see all the nitrogenous bases, and then you can see the daughter strands, which are gray, and they're being formed anti-parallel to each of the parent strands. Now, this is important because each of the parent strands is serving as a template for the daughter strands. It makes sure that our DNA replication is incredibly accurate. And that's what you want. You don't want to introduce a lot of mutations because of an inaccurate copying. Okay. Now, anytime I show you something simplistically, you should be going, how is the cell doing that? Let's add more detail. Okay. One of the most important molecules to exist on this planet. Wait, I know there's lots of important molecules that exist on this planet, but this large protein complex is called DNA polymerase. There's actually several different types of DNA polymerase. They each do something slightly different. I know they're adding DNA, but let's take a look at that word, polymerase. ASE tells you it's an enzyme. We got a chemical reaction going on. Making DNA is a chemical reaction. Poly means many. So DNA is made up of building blocks. You know them, adenine, thymine, guanine, cytosine. So DNA polymerase sticks the nucleotides together to form DNA. So hence the name DNA polymerase. Now, interestingly, remember I told you there's directionality to our DNA. DNA polymerase can only add a nucleotide to the 3' end. So it can't add it up there with a phosphate group. It can only add it to the 3' end, so the bottom of the ribose sugar. Now, you're probably also wondering, well, how does replication even get started? And how do you replicate 3.5 billion base pairs rather quickly? Well, it turns out that our DNA has what's called an origin of replication. These are sequences of DNA filled with A's and T's. Why would it be filled with A's and T's? Sounds like AT. All-terrain attack transport? Isn't that Star Wars? No. Adenine and thymine have 2 hydrogen bonds, right? So it's easier to pull apart adenine and thymine rather than a bunch of guanine and cytosine, because guanine and cytosine have 3 hydrogen bonds. So we might have 10,000 of these origins of replications throughout our DNA, allowing us to easily pull it apart. Now we call this non-coding. What that means is those A's and T's are there just to allow a protein to get in there and pull it apart. They're not coding for proteins. Oh, is it getting more complicated? Of course. There are lots of proteins involved with DNA replication. It's complicated. So first of all, how do you start to unravel your DNA? Well, you have something called a helicase. That starts to help pull it apart. Then, the DNA wants to come back together. Remember, the strands are held together by hydrogen bonds. Run your hand through water. The water comes right back behind it. You pull apart the DNA, it comes right back. So we got a protein called helicase, which pulls it apart. Then we have these single-stranded binding proteins. Single-stranded. So they're little proteins that will latch onto the DNA and keep it from coming back together. No, wait, isn't DNA a helix? It's twisted? So you ever taken rope and unpulled it? It all gets all twisted up downstream? So there's another protein called topoisomerase that basically keeps the DNA from getting all bound up and kinked up downstream. It cuts it and keeps it going and then puts it back together again. I know, interesting, isn't it? There's also things like DNA ligases. And then there's another problem. I told you earlier that DNA polymerase can only add nucleotides, you said it, to the 3' end of a DNA molecule. Well, if you pull this thing apart into DNA molecules, how does it work? Well, it doesn't. So you have to have what is called an RNA primase. Primase. An RNA primase actually goes along and adds a little RNA primer. And then DNA polymerase will start adding on the nucleotides to the 3' end. Oh, wait. We got these replication forks. We open up our DNA like this, right? Oh, let's try and get the camera there. We open up our DNA, and the replication goes this way and that way. But the problem now is I've got both my strands serving as parent strands. So now you've got a leading and a lagging strand. So this parent strand that's the leading strand is going 3' to 5'. So when I make my daughter strand, it's 5' to 3', so I can make it continuously. Now the lagging strand, the parent strand is in the direction of 5' to 3', but the daughter strand, wait a second, 3' to 5'. No, that doesn't work. You can't add nucleotides to a 5' end. So what happens is, as you've got the replications going this way, right? You've got the leading strand where it's just continuously replicating. Then you had the lagging strand where the DNA polymerase goes this far, pops off, comes back and does a little bit more, comes back and does a little bit more, and you create all of these fragments called an Okazaki fragment. And guess what? You need another protein to stitch all those fragments together on the lagging strand called DNA ligase. So as you can see, DNA replication is pretty wild stuff. It gets a little complicated. And then here's our summary, right? You've got your leading strand and your lagging strand. The helicase opens up the DNA at the origin of replication. Then the DNA polymerase continually will synthesize the leading strand going 5' to 3'. You got the Okazaki fragments, which are formed there on the bottom. And these are on the lagging strand. And of course, that's the DNA ligase, which is going to stitch all those together. And then that'll make one continuous piece of DNA. And then you've got your two daughter strands that are formed. So it's pretty interesting. Now, why did I go into all this detail about leading and lagging strands? That might be something you would find in a majors biology course. Well, there's a couple of reasons why I talked about it. One is I just want you to get an appreciation for the complexity of something like DNA replication. There's multiple proteins involved, like the DNA polymerase, isn't it? There's even DNA polymerase 1, 2, and 3. There's several different types of RNA polymerases. There's these ligases, topoisomerases, just all kinds of molecules involved with it. And they each have a slightly different function. So just to understand the beauty and the complexity of what's going on inside of our cells. Next, this has real-world implications for us. Remember that our DNA is linear. The two ends aren't connected. And we've got this leading and lagging strand. On the leading strand, I can replicate the DNA all the way to the end. But on the lagging strand, I cannot. That's a problem, right? So every time you replicate your DNA, you don't replicate all the way to the end. That means every time a cell divides inside of our somatic cells, our body cells, the cells of our skin and those epithelial cells, every time they replicate, the DNA gets shorter and shorter and shorter and shorter and shorter. Now, we think that that's an evolutionary adaptation against cancer because that immediately puts a limit on the number of cell replications they can undergo before you run out of DNA and you start chopping into your actual coding regions. Now, we don't want that to happen. So at the end of our DNA, we have these caps. And these caps are called telomeres. So my telomeres are probably shorter than many of your telomeres because I'm twice as old as most college students. So as I've aged throughout my life, my telomeres have gotten shorter and shorter and shorter. Now, even though I'm a little bit older than some of you guys, I just had a kid. She was not born old. She was born at age 0 and will age normally. And the reason why is because in our gametes, we have an enzyme, telomerase. And telomerase, what it does is it adds telomeres to the end of your DNA and kind of resets the clock a little bit. And in fact, in our cancer cells, some cancer lines are immortal. They keep going on and on and on and dividing over and over and over again. And one of the mutations inside of cancer cells is that in you and me, our telomerase is turned off in our somatic cells. But in these cancer cells, telomerase is turned on. So every single time the cancer replicates itself, guess what? It replicates with the full amount of telomeres and the cell clock is reset each time. So cancer cells may be immortal. So understanding that is important for treating cancer, but we also may learn how to treat aging if we understand the role of telomerase, how to turn it back on, and how we might reset the clock inside of our cells. All right. Well, that's been another episode. I hope you enjoyed it.